This lecture video is going to go over the places in our body that holds fluid and the different kinds of pressures that can cause fluid to move from one compartment to another compartment. So we're going to start with just a reminder of the types of fluid that's found in our body, or the compartments of fluid that's found in our body. So there's basically three different compartments. Really, there's only two that we're concerned about, but we throw that third one in there just as a reminder. But the first is going to be the intercellular fluid, or the ICF. So this is going to be all of the fluid that is found inside of our cells. So mixed in as the cytoplasm, all of that fluid. It is the biggest compartment or holder of fluid in our body. 67% of all fluid is found inside of our cells. Then we have the extracellular fluid, or the ECF. This is the fluid that is found outside of the cell, and we further divide this compartment into two categories. The interstitial fluid, which is going to be the fluid that's located outside of the cell and outside of the blood vessels. So this also includes the fluid in your lymph. And then blood plasma. So that's going to be the fluid that is included within the blood vessels. And that's going to make up, again, 26% of the total amount of fluid found in your body. So it's a much smaller compartment, much smaller amount than the intracellular fluid. And then last, there's the trans fluid, which is going to include all of the fluid that kind of sits in between epithelial cells, and in smaller compartments, it makes up less than 1% of the total amount of fluid. This would include your cerebrospinal fluid, your aqueous humor, your vitreous humor, those types of things. So it's pretty negligible, so we don't really talk about it. It doesn't really play any kind of role in fluid movement, but it is worth mentioning that there is this 1% of fluid that's not technically extracellular fluid and not technically intracellular fluid. So I have this handy dandy diagram that I attached, my beautiful handwritten or hand-drawn diagram. You can see artistry is not my strength, but again, it just is a general picture to kind of help us follow along with what's happening. So you can use it as you want, or you don't have to use it if you don't want to. But just to kind of orientate ourselves again with the different types of fluid and where they are. So here's a capillary bed, right? So here's the arterial end, here's the venule end. All right, so this is going to be the blood, this is a capillary. So a blood vessel, okay, and the capillary bed. And so, you got blood that's going to be floating out through here, or fluid. Up here is a cell, okay, so we know that inside of the cell is going to be the intercellular fluid. Okay, so here's a cell, here's a cell, here's a cell, there's cells floating around. We also know that then in here, we're going to have plasma, right? So this is extracellular fluid in the plasma compartment. And then we're going to have interstitial fluid floating all around. So we have interstitial fluid that's floating all around the outsides of the cells and outside of the blood vessels. And that'll also include the fluid inside of the lymph. So hopefully that kind of helps you see. What we're going to be adding to this diagram is how fluid moves from each of these compartments. How does it go from in the cell to the interstitial fluid? How does it go from the plasma to the interstitial fluid, or vice versa? So really the question is that as blood starts to flow into the capillaries, on that arterial end, some fluid is going to get filtered out, right? There are these little channels that exist in those capillary beds, and we're going to get some fluid that's going to filter out of the capillaries, right? Out of the blood vessels. Now, we don't normally want that fluid to stay out here in the interstitial fluid area, right? We don't want to have that, because think about what's going to happen if we get a whole bunch of fluid building up in this interstitial fluid area. We're going to end up with swelling, or we're going to end up with edema. Edema is caused by an accumulation of fluid in this interstitial fluid. So in order to prevent that from happening, the body is going to work really hard to basically reabsorb that fluid back into a different compartment, depending on the needs of the body. So the question is, and the question that we're going to look at is, first of all, why was the fluid forced out in the first place? Like, why did the fluid that was within these capillaries, why did they go out anyways in the very first place? And what kind of pressure drives the movement to not only move the fluid out of the capillaries, but then to either bring it back in or to move it to one of the other different compartments? And that's what we're going to focus the rest of this video looking at. And that's going to bring us to looking at two types of pressure that drive the movement of fluid from one compartment to another. So we're going to start with hydrostatic pressure. Hydrostatic pressure is a push pressure. It's a pressure that builds up and it will push the fluid from one compartment to another. So, think about if you have a water balloon and you hang it up to the faucet and you pump water in there. At some point, the pressure of the water outside on the outside of the balloon is going to be so high that the balloon's going to break and the water is going to go everywhere, right? Well, this is kind of like hydrostatic pressure. It's basically a buildup of pressure that's going to cause fluid to get pushed from one compartment to another. There are two types of hydrostatic pressure that we talk about. Blood hydrostatic pressure. This is going to push fluid from the blood vessels to the interstitial fluid, and this is caused purely by blood pressure. And then interstitial fluid hydrostatic pressure, which is going to push fluid from the interstitial fluid into the lymph. And this is going to be caused by changes in fluid volume. So again, if you look at the diagram, as the blood is coming down through this area and the blood pressure, the capillaries are getting smaller and smaller and smaller, narrower and narrower and narrower, the blood pressure is going to increase, right? And what happens is the pressure of that blood puts pressure on the walls of these arterioles. And that pressure pushes the fluid out to the interstitial fluid. So this here, going from plasma to interstitial fluid, is going to be the blood hydrostatic pressure. That's what's going to cause it to occur. Now, as the fluid builds up, and you can write another one of these down here, right? So hydrostatic pressure. So as the fluid builds up in this interstitial space, it's going to cause the fluid to go from the interstitial fluid into the lymph. Again, because the pressure, that water is putting pressure on these vessels and it's forcing the fluid then into the lymph. And this would be called interstitial fluid hydrostatic pressure. So this up here is going to be caused by the blood pressure, the pressure of the blood flowing through the arteries. As it gets pushed out, as the fluid gets pushed out into the interstitial fluid, it's going to then build up and cause pressure that will push the fluid then into the lymph vessel. And then the lymph is going to drain that fluid. So those are the two types of hydrostatic pressure. So if you have a push pressure, you have to have a pull pressure to kind of counteract that. So if you have a whole lot of solutes in the fluid, then there's going to be less water molecules. All right, so let's just imagine that you have a cup, beaker that has 10 milliliters of fluid in it. All right, the more salt that you have in that fluid, so let's say it's salt water, the more salt you have in that fluid, the less water molecules there are. The fewer solutes you have in the fluid, the more water molecules you are. And this is going to be what is going to drive the pull pressure, which is called osmotic pressure. It's going to be driven by solute levels that will then pull water from one area to another, from an area of high concentration of water, which is going to equal low concentration of solutes, to high concentrations of water. To low. So osmosis is pulling water from high concentration to low concentration, water concentration. Now, if you have a high water concentration, that means you have a low solute level. So really, the movement is going to be water, but it's going to be driven by the solute concentration. So membrane permeability is going to be a huge factor in this, especially in our fluid compartments, because this is the idea that the membranes, the plasma, the vessel membranes, the cell membranes, that they can let certain solutes pass through, which can allow them to kind of even out the osmotic pressure, but they don't allow all solutes to pass through, in particularly proteins, because proteins are so big. And so by looking at the amount of proteins and by looking at the amount of solutes that are present in any given fluid, we can determine what direction the water is going to get pulled. There are two types of osmotic pressures that we're dealing with. Blood colloid osmotic pressure, and this is going to pull fluid from the interstitial fluid into the blood vessel, and it's going to be caused by solute or protein concentration. And then interstitial fluid osmotic pressure, which is going to pull fluid from the cells to the interstitial fluid, and this is also caused by an increase in solutes. So again, if we kind of look at our diagram here, we can look at interstitial, or we'll start with blood colloid osmotic pressure. This is going to pull pressure from the interstitial fluid to the blood. So we're going to go into this direction and it will pull it into the venule end of the blood. So this would be BCOP, blood colloid osmotic pressure. And it's going to be, again, caused by the solute concentration of protein. So water will end up moving from high concentration here, which means there's fewer proteins on the outside of the vessel than there is on the inside. So there's more water on the outside than the inside, and so the water will pull into the capillary. And then the last movement is going to be the interstitial fluid osmotic pressure, and that's going to pull fluid from the cells into the interstitial fluid. So this is the interstitial fluid osmotic pressure. And it is also going to be driven by the concentration of solutes. So, I mean, you could kind of draw solutes right here and solutes inside of here. You know, water will get pulled, so from areas of high concentration to low, so high concentration inside of water to low concentrations outside of water, okay? So it would be opposite for solutes. So these are the four kinds of pressures then that are going to occur that will move fluid from the different compartments. So the two kinds of hydrostatic pressure and then the two kinds of osmotic pressures. Now, if you kind of wanted to summarize this and kind of look on this end, on the arterial end, you're going to have a very high hydrostatic pressure. And you're going to have a really low osmotic pressure. So fluid will end up getting pushed out. Fluid goes out. And on the venule end, it'll be the opposite, right? You're going to have a really high osmotic pressure, but you're going to have a low hydrostatic pressure, and that's going to allow water to be reabsorbed. So fluid will go in. So this week when we start talking about how we specifically monitor blood volume, it's important to have this general idea of the pressures that are involved in pushing fluid from one end to another, because fluid movement ultimately is going to drive or be driven by blood pressure, which is in part driven by the volume of blood and the volume of fluids within the system.